So welcome to another lecture of Biology 1030, Foundations of Cell and Molecular Biology. This lecture is going to talk about DNA replication and its applications. So there are a number of textbook readings and other resources that are available in Blackboard. You should be sure to read Chapter 12, Section 1, 3, and 4. Originally 12.4 wasn't in the required readings on Blackboard, but it is there now, so you should make sure that you give that section a read. There are also some additional Launchpad learning exercises and study tools available to you. Of course, you need to have purchased the Launchpad resource to be able to access those, and they are entirely optional, but they are available through Blackboard, or you can log in directly through the Macmillan site. So the core concepts of this chapter and the sections that we are covering is that with DNA replication, a single parental molecule of DNA provides two daughter molecules. Also, the techniques for manipulating DNA follow from the basic structures of DNA and DNA replication. And finally, that genetic engineering allows researchers to alter DNA sequences in living organisms. So at the end of this chapter, you should be able to describe the process of DNA replication, define the key terminology used in DNA replication, explain the process of polymerase chain reaction, interpret the results from gel electrophoresis, and explain processes of DNA modification and how these processes influence our daily lives. So, one of the primary premises in biology is that all cells come from pre-existing cells, and unless we want a bunch of empty cytoplasmic shells, the creation of new cells requires the replication of DNA so that DNA can be present in all of the daughter cells. So DNA replication is this process of duplicating the DNA molecule. Each parental strand is a template for a daughter strand. Also, DNA replication is semi-conservative, and that means that the new double strand is created from a template or conserved strand and a daughter strand which is generated. And understanding these molecular underpinnings of DNA replication is fundamental to some of the applications we use in things like genetic engineering and DNA research. During DNA replication, the two strands of parental DNA separate, and each parent strand serves as the template strand for the synthesis of a daughter strand. The order of the bases in the template strand determines the order of the complementary bases added to the daughter strand. And remember that A always pairs with T and G always pairs with C. So, if we were going to have a particular sequence starting at the 5' end that included the following bases. Say we have A, T, G, and C. Then the replication would occur in the complement as T, A, C, G. So originally there were two hypothetical models for DNA replication. In 1958, it wasn't really known how DNA replication occurred, and there were two common hypotheses for how it occurred. There was the semi-conservative model and the conservative model. So the conservative model suggested that after DNA replication, the new DNA duplex consisted of two newly synthesized daughter strands, leaving the parental duplex completely intact. And you can see in this particular situation, you've got this parental strand, and if it was totally conservative, you would just have replication occurring, and the parental format would remain throughout the replication on the one side of the replication model. The semi-conservative model suggests that after DNA replication, the new DNA duplex consists of one old strand, which is the parental strand, and one new strand, which is the daughter strand. So in the semi-conservative model, you have the parental molecule. And if it was going to be replicated in a semi-conservative way, it just means that, you know, part of the parental strand is maintained in the daughter strands. You would get half of it remaining in the replicated molecule here and half of it remaining in the replicated molecule here. And of course, in the second round of replication, in the semi-conservative model, you would have this pattern emerging. And conversely, in the conservative model, after the second round of replication, you would have this model emerging. So the question became, which of these models is in fact true? So Messelson and Stahl set out to answer this question, and their original experiment provided support for semi-conservative replication, but they only studied replication in bacteria. So DNA replication in eukaryotes, could have been different. So eventually there was experiments done with fluorescently labeled DNA, and that led to the discovery that eukaryotic cells also replicate semi-conservatively. So the cells were allowed to grow in a medium with fluorescently labeled nucleotides, and after 2 rounds of replication, the resulting chromosome was photographed at metaphase, because remember that chromosomes align in the center of the cell during metaphase, and they're condensed, so they would be very readily seen. If they were sort of disentangled and very loose, it would have been very difficult to see them. So you can clearly see that one chromatid containing the hybrid DNA, so they had the fully labeled, fluorescently labeled nucleotides, it fluoresced rather strongly, whereas the other side that only was half labeled only fluoresced partially, and that provided evidence for semi-conservative DNA replication. Let's review the structure of DNA. We're going to talk a little bit more about this when we move on to other chapters that we're exploring in the course. But DNA is a double-stranded molecule, and its strands are anti-parallel. So what does it mean to be anti-parallel? Well, that just means that the orientation of the molecule is on each side opposite. So here you can see this is the 3' end of the one side of the double helix, and you could follow that down and around here. And the other side is flipped and reversed to that. So it's anti-parallel. It starts at the 5' end and it weaves its way around down here to the 3' end. So the purine base in one strand pairs with the pyrimidine base in the other strand, and the backbone of DNA is composed of these alternating sugar and phosphate molecules. And the bases project into the center of the double helix. There is this major groove, which is just a larger section in the double helix that has this groove, and then the minor groove where they appear to be closer together. So adenine and thymine, which pair together, are held together by 2 hydrogen bonds, but guanine and cytosine are held together by 3 hydrogen bonds. So DNA polymerization occurs only in the 5' to 3' direction, and it's catalyzed by DNA polymerase, which is an enzyme. So DNA polymerase is a really conserved enzyme across different species, and its basic function is to synthesize a new DNA strand from an existing template. And most DNA polymerases also correct mistakes that happen in replication. So here we're looking at the template strand, and the daughter strand. And you'll notice that the daughter strand is in the 5' to 3' direction, whereas the template strand's in the 3' to 5' direction. So DNA grows in the 5' to 3' direction. And what's happening here is that incoming nucleotides are accepted if they correctly base pair with the template. So you see here that G is pairing with C. Note that this hydroxyl group is on the 3' end of the growing DNA strand, and that's where the nucleotides are added onto, and that's why it only grows in the 5' to 3' direction. Because that 3' hydroxyl group, or this OH, which is a hydroxyl group, of the growing strand attacks this high energy phosphate bond of the incoming nucleotide, and it cleaves it here and initiates the synthesis reaction to add on this nucleotide to the growing strand. Since newly synthesized DNA can only be elongated at the 3' end, the two daughter strands are made a bit differently. One strand, called the leading strand, has its 3' end pointing towards the replication fork. So you can see this 3' end here. Here, let me get the laser pointer, see if that helps us. We see the 3' end is pointing towards the replication fork. This is the replication fork where the DNA is separating. And we can see that it can attach its RNA primer, which starts the process of replication, and just keep adding nucleotides on all the way along the leading strand as the DNA unwinds. But on the other strand, the lagging strand, this isn't actually possible because it starts with its RNA primer at the 5' end and then has to go to the 3' end, which causes it to sort of hit the end of the DNA strand quickly, and it has to jump back and do it in more piecemeal type of fashion. So the leading strand has continuous replication and the lagging strand has discontinuous replication. So this fragmented replication on the lagging strand ends up being these pieces along the lagging strand, and the leading strand has this continuous replication. This requires the replication from the lagging strand to have some kind of repair happen because we've got these RNA primers all along the newly synthesized strand. So they need to be removed and replaced with DNA. And the fragments of the lagging strand then get ligated after removal of this lagging strand together so that we end up with two continuous strands of DNA from the leading and the lagging strand. So the lagging strand just after the replication occurs needs a bit more post-processing. You may find in your textbook that these sections that are replicated from the lagging strand are sometimes referred to as Okazaki fragments, and that's named after the scientists that discovered them. DNA polymerase can't begin a new strand of DNA on its own. It can only elongate the end of an existing piece of DNA or RNA, so that creates a bit of a problem. Each new DNA strand has to begin with a short stretch of RNA that serves as a primer for DNA synthesis. So it needs to be primed in order for the polymerase to actually work, the DNA polymerase to work in the elongation and replication of DNA. So the primer is made by an RNA polymerase called RNA primase. So the RNA primase synthesizes this little short piece of RNA that's complementary to the DNA template. And then once the primer has been synthesized, the DNA polymerase takes over and elongates the primer, adding successive DNA nucleotides to the 3' end of the growing strand. So here you could see the RNA primase, protein that's attaching this RNA primer onto, in this case, the lagging strand. And then the DNA polymerase attaches on the complementary nucleotides in the 5' to 3' direction. So the RNA primase is responsible for laying down the RNA primer, and the DNA polymerase extends that RNA primer with nucleotides that are complementary to the template strand. As a result of this system, all new DNA strands have a short stretch of RNA at their 5' end, even the leading strand. The lagging strand has a lot of little short RNA pieces, one for each fragment. When the growing fragment comes into contact with an RNA primer, as you can see that's happening here, so you could see this RNA primer was actually here, and this growing strand hits it. What happens is that a different DNA polymerase complex takes over and it removes that RNA primer, replacing it with DNA nucleotides. So when the replacement is complete, an enzyme called DNA ligase joins the fragments together so that we have one continuous daughter strand, and that the daughter strand from the leading strand and the lagging strand are equally continuous.